Hello friends, today we are going to study about protein function, that is reversible binding of a protein to a ligand, part 4. In this part, let's first understand the hemoglobin quaternary structure. Now, as we already know that hemoglobin consists of 4 subunits, that is 2 alpha subunits and 2 beta subunit. In alpha we have alpha 1 and alpha 2. In beta we have beta 1 and beta 2. Now these 4 subunits are arranged in a way like this. That means you can see here alpha 2 and beta 2, and the second half is alpha 1 and beta 1. So these are the 4 subunits. Now because of this we say that hemoglobin is a tetramer because it has 4 subunits, or it is a alpha beta dimer. That means it has 2 parts, each half consists of 1 alpha and 1 beta subunit. Now the highlighted portion, as you can see, these are the interaction between these subunits. The alpha 1 beta 1 subunit, that is these interactions which are highlighted. Similarly, the alpha 2 and beta 2 subunits, these interactions are very strong, and these interactions, that is the interaction between alpha 1 beta 1 and alpha 2 beta 2, these interactions involve 30 residues, and these are very strong interaction. Whereas the interaction between alpha 2 and beta 1 and alpha 1 and beta 2, these interactions involve 19 residues. So what are these interactions? One interaction is between alpha 2 beta 2 and alpha 1 beta 1. These are strong interactions involving 30 residues. And the interactions between alpha 2 beta 1 and alpha 1 beta 2. These interactions involve 19 residues. Now the interaction between alpha 2 beta 2 and alpha 1 and beta 1, these interactions are so strong that if we treat the hemoglobin with urea, the tetramer will disassemble, but these interaction will remain assembled even after treatment with urea. And the interactions between these subunits, they involve mainly hydrophobic interactions. Apart from that, we can also see hydrogen bond interaction and electrostatic interaction. So hemoglobin quaternary structure involves 4 subunits, alpha 1, alpha 2 and beta 1 and beta 2. The interactions between alpha 1, beta 1 and alpha 2 and beta 2 involves 30 residues, and these are so strong that even after treatment with urea, these dimer remain intact. Whereas the interaction between alpha 2 beta 1 and alpha 1 beta 2 subunits, these subunits involve 19 residues. Now, binding of oxygen cause structural change in hemoglobin. Hemoglobin has two major conformations. These are called T state and R state. T stand for tensed and R stand for relaxed. This R state has high affinity for oxygen. Therefore, binding of oxygen stabilizes R state. That means what we can say? This R state is a state of oxyhemoglobin. The hemoglobin bound with oxygen. Now the reverse is true for the R state. It has low affinity for oxygen. Therefore, absence of oxygen will stabilize this structure. That means what we can say? This state is of deoxyhemoglobin state. Now this T state, the state is stabilized by various ion pair interaction or electrostatic interaction. The amino acid involved in this ion pair interaction can be seen here in this diagram. So these are the amino acids that are involved in the ion pair formation, stabilizing the T state. With the T state, as soon as an oxygen will bound, the T state will convert into R state. During this conversion, because T state is stabilized by some ion pair interaction, so during this conversion or transformation, the existing ion pair interaction will be broken down and some new interactions will form. Correct? Because the state is now changing from T to R. So T state is stabilized by some ion pair interaction. So to convert from T to R state, those ion pair interactions should be broken down. And when the R state is formed, this for the stabilization of this state, some new ion pair interaction will form. Now, This how the T state and R state looks like. This is T state, that means stabilized in the absence of oxygen. This is R state, which is stabilized in the presence of oxygen. Now, when this conversion is happening from T to the R, because various ion pair interactions are getting broken down and some new interactions are forming, so during this transition, the individual subunit changes a little. And what happens is the alpha beta subunit pairs will slide past each other, due to which what happens is you can see the gap in the center in the T state, and here the gap in the center in the R state. So what is happening? The gap is narrowing. Why? Because one alpha beta pair is sliding over the another alpha beta pair, due to which what happening is the gap present between these subunits, they get narrowed. So this is the difference between T state and the R state. Now, when this conversion is happening, if we see little deeper, then we can find that in the T state, the Fe2 plus is 0.6 angstrom out of the heme plane. When the transition is happening, this Fe2 plus will move into the center. Where this Fe2 plus is present is associated with heme, and where the heme is present inside the pocket of globin. This we already studied. Now, when it is present inside the pocket of the globin, only oxygen can bind, because the binding of other molecules like CO2, H2S, etc., they cannot bind due to steric hindrance. Now, in the T state, when Fe is not bound with oxygen, it is little bit outside the heme plane. As soon as the oxygen will bind, the Fe2 plus will move into the center, into the pocket. Next, what will happen? The F helix will tilt. In the diagram, you can see here in the R state, F helix is there, to which this histidine is attached. In the last video, I asked you to remember two important positions, that is his F8 and his F7. These are the amino acid positions that are attached with Fe. Histidine F8 is directly attached with Fe, whereas histidine F7 is not directly attached with iron. It is attached with oxygen, the oxygen attached with iron. So that's why histidine F8 is known as proximal end or proximal histidine site, whereas histidine F7 is the distal side. So as soon as the oxygen is bind with the Fe, it will move inside the center. Then next what will happen? F helix will tilt a little. Because of this tilting, there occur a movement in alpha 1 beta 2 and alpha 2 beta 2 interface, which will break the ion pairs, and hence the T state will transform into R state. So, and for the stabilization of R state, some new ion pairs will form. Hemoglobin binds oxygen cooperatively. As we now know that hemoglobin exists in two states, that is T state and R state. T state means deoxy state and R state means oxy state. Hence R state has high affinity for oxygen and T state has low affinity for oxygen. Now what happens is, suppose these are the four subunits of hemoglobin in T state. As soon as the oxygen will come, the first oxygen molecule, it will come and bind with the T state. So what will happen then? State will convert into R state. Now, as soon as the T state will convert into R state in one subunit, the message will communicate it from the converted or the transformed subunit to the subunit which is still in the T state. Now, this message is going to communicate through subunit-subunit interaction. Subunit-subunit interaction, the message will be communicated to the another subunit. Now, what will happen? As the message is communicated, this subunit will convert into R state, and because of which now the binding of second oxygen molecule will be with higher affinity compared to the first one. So this how the binding of first oxygen molecule will affect the binding of subsequent oxygen molecule, because as soon as the one subunit will get converted into R state, that is the higher affinity state, that message will be conveyed to the next subunit through subunit-subunit interaction. Hence, the hemoglobin have sigmoidal curve. Now next is Allosteric protein. What are allosteric protein? These are proteins in which binding of a ligand to one site affects the binding properties of another site on same protein. So what it is saying? What are allosteric protein? These are protein in which binding of a ligand to one site will affect the binding of another ligand to another site on the same protein. So those proteins are known as allosteric protein. Now such ligands are referred to as modulators because they are modulating the binding properties of another ligand. So these modulators can be either inhibitors or activators. That means the binding of first ligand can increase the affinity of the second ligand or it can decrease the affinity of the second ligand. Now basis on this, there are two types of allosteric protein, that is homotrophic and heterotrophic. Homotrophic means sometimes what happens is the ligand and the modulator, these are separate molecules. When the ligand and modulator are the same molecule, then we call it as a homotrophic. When ligand is a different molecule and modulator is a different molecule, then in that case, we can call it as heterotrophic. So in case of hemoglobin, what happens is protein is hemoglobin, ligand is oxygen. So here oxygen is a ligand, but binding of one oxygen affects the binding of another oxygen. So here oxygen also acts as a modulator. So because the oxygen itself is a ligand also and a modulator also, therefore hemoglobin is a allosteric protein. Now this subunit-subunit interaction, how it occurs? Whenever the oxygen bind to one subunit, this subunit will change its conformation. Now this change in conformation will then mediate it to another subunit, making this subunit more compatible for the ligand. Now for this, each subunit can have multiple segments. Now some segment can be stable, some segment will be unstable. So in each subunit, there will be stable segment also, there will be unstable segments also. Now, when the ligand will come and bind to the stable subunit, what will happen? This message will transfer to this unstable subunit, making this unstable subunit stable. This how all the unstable segments of a single subunit will convert into stable segment. Once this conformation is done, this whole subunit is now having different conformation. This message will then transfer to its neighboring subunit, making it more stable or giving its higher affinity for the ligand. So two models has been proposed for the cooperative binding. The first model is symmetry model, or which is also known as MWC model, from the initials of the scientist who proposed this model. The model is also known by all or none model. What the model says is all the subunits of a protein will either be in T state or it will be in R state. Now, these two states will be in equilibrium with each other. To T state, the ligand will bind with low affinity. To R state, ligand will bind with high affinity. Now, subsequent binding of ligands, like for example, here one ligand bind, in the next two ligands bind, three ligands bind. So, the subsequent binding of the ligands will change the conformation from the low affinity to the high affinity conformation. And the next model is sequential model. According to sequential model, the subunits will be in T state. The binding of ligand to one subunit will convert it into R state, that is the high affinity state, and this change in conformation will then communicate it to the neighboring subunits. Like you can see, these two subunits are the neighboring subunits, so their shape changes a little. Now, the binding of second ligand to the second subunit will change the shape of the subunit neighboring to that subunit. So likewise, when the ligands are getting attached, the subunits are getting converted from T state to R state. Whereas in symmetry, what it is saying, all the subunits will be either in R state or it will be in the T state. If it is in the T state also, it will remain in the T state, but in equilibrium with the R state. So more and more binding of ligand in T state, which is a low affinity, will shift the equilibrium towards high affinity conformation. Now, one question from my end for you all, that is, if all the subunits of hemoglobin are separated, how would it affect the oxygen binding curve? Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.